Okay. Connor told me that my intro is lackluster, I would say. So,
True.
Welcome to Unsupervised Thinking, a podcast about neuroscience, artificial intelligence, and science more broadly. I'm Grace.
I'm Josh.
And I'm Connor.
And the topic for this episode is optogenetics, which is fun, and so we're going to talk about it and what it is and how it got started and what it means to everyone today. Because if you have worked anywhere near neuroscience in the past 10 years, you've heard about the joys and wonders of optogenetics and its miraculous abilities to save us all.
Wow, that sounded very sarcastic. But yes, I actually agree, Grace, that you did previously sound, I wouldn't say lackluster, but you sounded like a serious news broadcaster introducing something terrible.
Well,
ironically,
we are more serious newscastery today because we have very fancy recording equipment. So,
well, recording equipment technically is still a laptop, but we have. I guess this is recording equipment.
Yeah.
It's
nice
transducing our vocalizations into electrical impulses, kind of like.
I mean, basically, we're playing with some new equipment, and we will buy better equipment to make it so that we're not recording from a microphone laptop. Or laptop microphone.
Speak in the direction of your microphone.
Yes, I know.
And don't reveal our previous horrible amateurish techniques.
Yeah, you may have noticed that previous podcasts sound terrible, or you may have just not listened to them because they sounded so terrible. I finally convinced Josh and Grace that was an important factor, and so here we are.
Okay, optogenetics, right?
Right.
So broadly, it means light and genetics. But really what people in neuroscience talk about when they talk about optogenetics is this ability to stimulate cells using light. So you can make neurons active or inactive by shining light on them. And it's kind of complicated how we got to this point where we could do that. So we can go through some of the history of that. But that is the broad idea, and it's influenced a lot of things because it's made it possible to do experiments that you couldn't do using standard stimulation techniques, like just sticking an electrode in and pulsing current through. So that's why it's been exciting, and it has been exciting, so I'll stop sounding sarcastic about it.
So, I mean, basically, we're talking right now in 2016, early 2016, and in 2015, people were dating that, like the 10-year anniversary of optogenetics. So there were some articles that came out where people kind of, reviewed this, the developments and trends that have occurred since there was like a 2005 paper that kind of marks the beginning of use of optical stimulation of neurons to control their activity. And prior to that, people had figured out that there were proteins in algae, even decades earlier, that were photosensitive.
And there's proteins in our eyes that are photosensitive. So
sure, yeah.
Throughout biology, there's been discoveries of different organisms that have different types of photosensitive proteins.
But specifically, like photosensitive ion channels, right?
Right, yeah. So the thing that people talk about as being the revolution in neuroscience does really stem from this 2005 paper with Ed Boyden as the first author in Carl Dijsselroth's lab at Stanford. And there were techniques before that attempted to let neurons be controlled by light that were slightly different in style than the one that became the most popular.
Yeah, what's the difference actually? Like, because I thought in that Q&A that we read in the Nature Neuroscience thing, there was one of the guys who did those papers. He was the senior author, and he made it seem like they had put light sensitive ion channels into neurons before that paper.
So that was the big one that the 10-year anniversary was being celebrated for was in 2005, and in 2002 there was a paper published that had this setup which they were able to create an acronym called CHARGE that described it, and I kind of wish that was the one that had won, because then you would call the neurons that had this ability charged neurons. But things don't win in science for those reasons.
Sometimes
but yeah, so in that one, it was that you had something that was photosensitive that they got from the eyes of fruit flies, and it connected to this like G-coupled protein type thing that could activate cells.
So just to be clear, so cells, when they fire action potentials, fire because ions flow through channels, essentially, and
in their membrane.
In their membrane, and the G-coupled protein receptors are kind of an indirect mechanism, whereby receptors on the membrane are stimulated, and then kind of internal cellular processes modulate something about the cell. But like the ion channels themselves are usually not directly modulated in that case. You know, it's more indirect. And so the kind of optogenetics that we use these days are the ion channels themselves are directly stimulated by light. And so these kind of other mechanisms and the way the photosensitive pigments in like the eye or other kinds of settings work tend to be kind of more indirect mechanisms that like don't directly activate ion channels.
Yeah, so the previous method was a bit clunkier, and this method that took off, which uses Channelrhodopsin, is kind of sleeker and simpler, and it's a single gene that encodes it.
So, but these genes came from algae, right?
Yeah, it is I have the Latin name, Chlamydomonas reinhardi, something. It's a single-celled algae, swims with 2 flagella, found around the world, and it has this protein naturally occurring in it called Channelrhodopsin that it probably uses to sense how deep in the water it is, because light levels change with depth in water. And so that's where they found this very nice gene for this protein that they could then give to neurons, so that neurons could now have an ion channel that responds to light.
Did they find? No, people knew about this before.
So people knew about that for a while, and in the years shortly before it was this protein was expressed in neurons, it was expressed in other living tissue by people who also collaborated, it seems, on the work to get it expressed in neurons. So it was expressed in other organ tissue.
Yeah, so in this 10-year look back that we read in Nature Neuroscience, a lot of people were talking about how this idea was kind of in the air before it really, took off, and how people at various points had kind of considered this possibility of putting something that responds to light into neurons to be able to control their activity, but no one had the guts to do it. Or actually, it seems what really happened is that funding agencies wouldn't give you money to try it. So that's even what happened to the people who were successful. I think it's the case that they were rejected from normal grants to attempt this, and something called the Pioneer Award was what ultimately gave them the ability to do this, and that's a special type of grant that's supposed to be like high risk, high reward.
It's an NIH grant, right?
Yeah. So that's kind of interesting that this thing that now everyone cites as one of the most important things in neuroscience in decades and all of that, and it's so revolutionary, it could have very well not really happened because people were just too cautious, I suppose, about attempting it.
It's also interesting in that Nature Neuroscience kind of Q&A thing where various people give comments and answer various questions about it, about optogenetics. It was interesting the way a lot of these people who now use it a lot, presumably in their work, when they were asked if they knew or anticipated the huge effects that optogenetics would have when they first came across, say, that paper in 2005. And many of them were very skeptical and thought that it would, be very fiddly, not reproducible, not robust, that it would be very dependent on the parameters and so on, and the system that you used it in. Which is kind of interesting.
And it turned out to be quite robust, and that surprised some people. I mean, I think that I was a little skeptical when there were people who kind of offhand said that they thought it would definitely work.
Yeah. You think that was hindsight?
Yeah, that's a little bit of... It's 10 years later, you kind of forgot that you were skeptical of it in the front end, or it's the kind of person maybe who's enthusiastic about everything, and most of those things don't turn out to be as interesting.
Yeah, exactly. I thought that was funny because it's like, you could read that maybe as if you were not a practicing scientist, which I guess we kind of are now, you might read that and be like, oh, scientists are so cautious. Like, why are they always shutting people down who are innovators? But like, obviously, you're skeptical, and most of the time you're right to be skeptical, right? And then every so often you're skeptical and it turns out, okay, it actually worked really well.
Yeah.
But I think it's easy to kind of sell that kind of thing in popular science reporting as like, ooh, those silly, cautious, conservative
like
scientists.
The man holding everybody back, just trying to advance science. But yeah, they also, people were saying how it seemed like there was a role played by how open the lab who started this was to share the reagents and the techniques and all that kind of stuff. Because usually, it's not the case that you read a paper that has a new method and then you just go and do that method and it's so easy. You just follow the step-by-step directions. These things usually do require some fiddling and things that maybe they don't think to put in the paper but were actually important for the ease of use. So it seemed like the culture that was around optogenetics, especially when it first started, was very much to share things and help
still, I think.
out.
They still. So they seem like a very generous network of labs that make it very accessible for new groups that are like from credible places to easily get the.
And it could have been different because I think someone else who had thought of this idea beforehand said that they were seeking a patent for something about it. So people, different labs very much execute their ideas very differently, and so this one is special partly just because of how it was executed.
How does that kind of thing work? Like if someone discovered this and somehow patented it,
if someone modified it, so if they were using a tweaked version of the gene, then they could distribute it however they wanted, right? The patent would only apply to the specific first version. Isn't
it the case that, I don't know, but I thought it was the case that if it's not for commercial use, there's not much that a patent does?
Yeah, no, so a lot of none of us are so big into experiments, but a lot of reagents are purchased from big biotech companies, and they're sold, and a company could sell them, and if they didn't have a patent on it, other companies could sell them. So hypothetically, if this was a very useful tool that everyone wanted to use, a big biotech company could be selling the thing at like a profit, not just like the shipping costs, and that would, I'm kind of I think many scientists are skeptical of that in the context of science, because it seems like, there aren't that many scientists, like you're going to try to extract money from the scientists. Like, especially if, as a scientist, if it was, government funds that allowed you to do the research that you then want to patent, it seems a little unfair to many people in science, and potentially to taxpayers. I think they would be right to be a little outraged about that, because you were basically paid by the taxpayers to do your work, and you're trying to like get double paid for it in a sense by also patenting it, when instead you could be essentially sharing what you've been paid by the taxpayers to do and allowing other people to bootstrap off of that.
How does that work, actually? This is a real digression, but if you do a bunch of stuff in Stanford as a lab in Stanford or something, they kind of automatically have some rights, right, to products.
Things can be patented, right? You can.
No, I'm talking about the university.
The university patents. Yeah, I don't know all of the details about this.
Depends on the university.
If you do it at the university, will
have some rights to it.
Yeah, if you use their tools, then they own the patent, I think.
Yeah, I think so, right?
And they
it's really funny that doesn't apply to like the government. If the government gave you a bunch of money.
The government doesn't own the patent, but the university
have partial
ownership over the patents. Yeah, that's an interesting point.
I never thought about it before.
So we can go into a little bit more of the mechanisms of this. Basically, there's this ion channel, and it has this component that when activated by light so usually the light has to be of a certain wavelength or a certain small range of wavelengths. So what that means is that it's of different colors for us.
So, initially there was one Channelrhodopsin or whatever that people were using, and it was activated by a certain, band or color of light. And now people have genetically engineered a whole kind of suite of optogenetic tools. So different proteins that do slightly different things to the cell, like inhibit or excite the cell, and they're stimulated by different bands of light frequency, so like different colors of light.
And when the light hits it, there's this component called retinol, which is related to vitamin A, that causes a change basically in the shape of the ion channel that allows it to open and let ions of a certain type come through, usually ions that are either positive or negatively charged, which is important to activating cells because cells get activated when their voltages reach a certain threshold.
And this is essentially the simplest mechanism that like cellular biologists could conceive of to activate cells by light.
Just like to step back slightly, just to make kind of maybe to demystify something for people who don't know. Like I almost know nothing about it, but for people who know very little about, say, molecular biology or biophysics in this way, just like a protein is just a big, huge molecule, and an ion channel is a protein, and it's sitting embedded in the membrane of a cell. And so it is kind of just a very literal thing. There's some component of these opsins, the thing that Grace just mentioned, retinol, which absorbs light in the way that, you know, all molecules can to some extent. And it causes literally like a change in the shape
it's like
of
there's
the protein.
like the different parts of the protein pull on each other until the protein's in a different shape.
Yeah, and that different shape of the protein allows it to pass certain ions kind of through it. That's why it's called a channel.
Yeah, so
they can travel
into
across
or out of the cell.
Yeah, travel across the cell membrane.
And it was found that, so this, as we said, this depends on retinol, and it happens to be the case that in mammalian tissue, actually I think they said in all vertebrate tissue, I don't remember, there happens to be sufficient amounts of retinol for this mechanism to work. So it could have been the case that there wasn't enough retinol to make a bunch of cells in the brain be able to function like this that didn't previously.
Oh, I see. I didn't know that. So retinol is just like some molecule.
It's a molecule that gets embedded in the protein.
And it like binds to the protein.
Yeah.
So it's not part of the protein.
Yeah. Well, I mean, what does that mean?
Yeah. Okay, but it's not like
it's not genetically.
Yeah, it's not genetically encoded.
Yeah.
Yeah. So I think there was some fear that perhaps they would have had to throw in a retinol gene when they put in these ion channel genes.
Or something.
Yeah, or in animals that don't have enough, like in I think C. elegans, the little worms, they enrich their diet with it, I think, to make them have enough. So yeah, so I guess this is another reason why people were surprised by the ease of use of this, is because there are all these things that can go
Just
wrong.
Coincidences, like essentially there's a whole bunch of extra molecule that isn't being used natively in mammalian cells or whatever.
Though to be fair, I mean, they did obviously put a lot of work into actually making this work, and there were things that they had to engineer beyond the way the protein naturally was to make it really function.
Yeah, I mean, this was like a huge engineering effort to make all of this work so comfortably beyond the sort of basic innovation and the basic success.
So yeah, so they found this protein and they know the gene that codes for this protein, and so now they wanted to put it into neurons so that they could control the activity of neurons. And so there's basically two different ways that you can do that. One is to use a virus, because the way a virus works is that it puts its DNA into the host cell's DNA. And so if you have a virus that has the gene for the ion channel in it and you inject it into a cell, it'll put that gene into that cell and it'll start producing that protein, which is the ion channel. And then the other way is to genetically engineer animals to have that gene as part of their genome from the start. And usually it is placed in such a way that it kind of follows a gene of interest. So you could have the gene that marks all neurons and it doesn't show up in any other cells. So it's kind of like a marker for neurons in the body. And then you could have, following that gene, you'd have the gene for the ion channel, so that all neurons are then expressing this ion channel and other cells are not. So those are the two main ways that people do it.
And there are a few core animals like flies and C. elegans, the worm, and mice that we can actually genetically encode things from birth, and for many other kinds of animals that you would instead use a viral vector because we don't as commonly genetically engineer them.
Yeah, so there's a few different reasons why you might want to use one versus the other. The viral vector is kind of nice because you might not know all the genes that code for the specific types of cells you want, especially if you want to localize the cells that have this channel to a certain part of the brain. You might not know the genetic marker for cells in the fourth layer of this certain area of cortex you're interested in or something. So if you have the virus, you can inject it just into that area, and then you are spatially controlling the cells that end up expressing this channel.
But it might be less cell type specific.
Yeah, so it's complicated. You can do things to make it more cell type specific. You can genetically encode from birth something in the cells of interest that means that they are the only ones that will be receptive to the virus when you put it in. So that's a combination of kind of spatial and cell type specific. So there's a lot of different ways that you can do, and depending on what things people are studying, they're going to combine all different things to get the cells that they want to have the amount of virus that they want.
And briefly, maybe it's worth talking about like how well we can control this. So we can control a lot about like the spatial or cell type localization.
I think some people would say the cell type even. I mean, you have to know some specific marker.
Yeah. No, but so we
for the cell type you want, which isn't just neuron or not neuron. There's lots of different types of
course.
neurons.
Yeah. So we can do that fairly well. But it's my understanding is that like the quantity of protein that's expressed. This is more a comment about genetic engineering in general and less about optogenetics specifically. But like we kind of cause the cells to just overexpress this protein like kind of all the time. So it's like kind of chronically on, and at least that's generically what is often done. I mean, there is talk in one of the other papers we talked about these principles of designing interpretable optogenic behavior experiments, and other work where people talk about kind of the issues where like this, so like in the original paper, in the 2005 paper, they did test that like the cells didn't die after a week of expressing this original variant of the gene. But since then, we now know that on kind of longer timescales, there can be like build up to a toxic level of the proteins when they're expressing optogenetic indicators or proteins. Sorry.
So yeah, there's different complications due to experimental design limitations, and then also due to potential toxicity and not knowing exactly what cells you're hitting. And also in some situations, in some animals, you might get cells that are endogenously receptive to light levels that could be affected, or the heat from putting an LED into the head of the animal could affect it. So yeah, so the way that you end up stimulating these cells, you need to get light into the brain. So you usually, if you're doing kind of like a rodent study where the animal is running around doing a behavior and you want to stimulate certain cells, you cut a hole in the skull and you drop an LED into the area that has the cells that have the channel or the area of interest for you, and you turn that light on and off, and then that controls if those cells are being activated by the light. Or we didn't explicitly say, but you can also inactivate cells using different types of channels. So they just change the flow of ions in the other direction so that you're not exciting the cell, you're inhibiting the cell. But the idea is the same, and they use different light colors so that you can be turning on one population and turning off another by having different LEDs in different locations.
Sure. So Channelrhodopsin, right, uses chloride channels, right?
Yes.
Yeah.
So that's the different type of ion channel that lets chloride ions out of
into the cell
to inhibit it. So there's also some people say that it would be better if it weren't light, but different types of electromagnetic radiation that may be able to penetrate better, especially if you're going to use this in larger animals like primates. I think there's some people feel that there is some issue about the light penetration and also the spread of it. If you want to be able to target things very specifically, it might be better to not be using light.
And if that's the case, though, then you do want to wear like tinfoil hats.
I don't know if well.
I mean, if someone injects, optogenetic proteins into your brain, and they can stimulate them across the skull from a distance using like electromagnetic radiation, then we should all hide in the woods.
I feel like you're going to turn into an anti-vaxxer because you're going to think that they're genetically altering your neurons when they do it.
Oh my god, they might do that. That's crazy.
Now they will because we said it.
Yeah.
We just gave them that.
So yeah, the way to control a population of a country is going to be to, in their vaccines, surreptitiously inject them with. So inject surreptitiously with the vaccines a viral vector to selectively infect certain kinds of neurons that will be responsive to electromagnetic radiation transcranially.
Yeah. See, I would almost be happy if that were possible, because it would mean that we understood the brain a lot more than
No,
we do.
because you could do things like just put them to sleep, for example.
Okay, yeah, you could. We know enough about the brain to be able to put people to sleep.
That would be really useful. Imagine that for like riots, right? Instead of having like a water cannon, they just have like a big light. It's like,
No, but it's not even a light. It's
it'd be like it'd be like
some
a ray gun, X-ray, or whatever.
But aren't there a bunch of?
And everyone just falls asleep. Can you imagine that?
We have so much magnetic radiation around us already; it would really interfere. I think.
Yeah,
we'd have a lethargic population.
Yeah. It'd have to be like radio frequencies. Like there's a specific one that you have to be on.
Right. Yeah.
We should all go work for DARPA.
Yeah, we do well. God, that's creepy.
So we've talked a bit about what optogenetics is, kind of a little bit about its history, and how to get it into cells. So what are some cool things people have done with it?
Well, we can say generically, kind of, its advertised benefits compared to previous methods. Yeah. So one is, as we talked about, this cell type specificity that you couldn't get if you were just sticking electrodes in and stimulating any cell that came in contact or near the electrode. You couldn't, say, oh, I only want the inhibitory cells, or I only want the cells that express this protein. You couldn't do that kind of thing. The other thing is the temporal control, because it's light, so you turn the light on, you turn the light off, and especially in later models of this ion channel, it's been engineered to be very responsive, so it's kind of like as soon as you turn the light on, the cell might start firing, and as soon as you turn it off, it'll decay like really quickly.
Oh, yeah, and Dijsselroth, he came to Columbia last year and Carl Dijsselroth, the guy who was the lead author on the 2005 paper, or not the lead author, the senior author on the 2005 paper. They engineer like all these crazy new tools all the time. One of the really cool ones was that they had like a switching one where a pulse of light would switch its state somehow and it would just stay on. So you could like turn them on like for like ages, and then
I think a different frequency of light turned it off.
Turn it off. These kinds of things. Anyway, so they make all kinds of.
Yeah. So there's it's very common for people to talk about the optogenetic toolbox, because they've just created. Yeah. They've just created all these different kinds of things to do whatever you may want. So those are kind of the two main things that are really different from any previous techniques.
So like a previous very common technique for stimulating neurons, right, was just kind of electrical microstimulation, where you just sort of put electrical electrodes into some brain area and just run current of various kind of frequencies and such. And so like people do that, and there's been like many studies for decades doing that, I guess.
Also very useful.
And the
many discoveries. Temporal
control, is fine for that, but
it's like super, but like not even specificity at the level of like, cell type or.
It's just a whole big region of the brain, it depends on the size of it, but you can't target like a specific neuron easily unless you're patched to that.
You don't even really know what it does, right?
Yeah,
sure. Yeah.
Like if you had a single neuron, you could know, because you could like
you can stimulate a specific neuron with electricity.
Its membrane. But if you just like run current of different frequencies into a brain area, what exactly it does is probably
you could
quite
be
complicated.
Exciting inhibitory cells that actually inhibit things, or exciting excitatory cells so much that they're not firing anymore. And like it's And
there's similar problems, of course, with optogenetics. If you try to just say excite all of the excitatory neurons in an area, then those excitatory neurons will excite inhibitory neurons, which will inhibit those excitatory neurons. But like at least you kind of know where
that's what you're doing.
Yeah, at least you know where it begins. Whereas in the electrical case, it's sort of all kinds of things could be happening. All the same.
And so there were previous methods that allowed for cell specificity to some degree, including these like viral methods where you're targeting cells that have a certain gene in them and that kind of thing. But the temporal resolution for those was bad. So it was either you created a mouse line that just never had a certain type of cell to begin with and see what happens. And so that's like the lifetime of the mouse is the temporal scale for that, rather than just being able to shut off certain cells. Or you can inject like mucimol is a substance that tends to just shut down a lot of cells. So you kind of like turn off a brain area by injecting mucimol into it, but you're turning everything off there, so it's not so specific. And the time course is, like you can't just switch that on and off on the scale of seconds. That's kind of minutes to maybe even hours for the effect to wear off if you're using pharmacology in that way.
Yeah. And so there kind of there were also other techniques using microscopy. So like for a while, uncaging started being very popular. So the idea there is that there would be inside of a cell, you would have some chemical that, like a neurotransmitter, for example, that would activate the cell or deactivate the cell, and it would be bound to something, and then you could stimulate the thing that it was caged in. So the thing that would activate the cell was caged in some other molecule, and by zapping that caging molecule with light, you could release that the chemical that it had caged, and it would activate the cell or something like this. And so that was kind of another thing that you could do with optical methods as well. But I think now that's presumably, so people were using that for a bit, but I think that's less popular now because optogenetics is pretty useful. I don't know if people are still using that a lot. I think presumably there's a lot of flexibility there because you can use optics to control a specific chemical there using microscopy.
I think like people still use uncaging for like super spatially specific things sometimes,
sure.
Right? Like uncaging like on certain parts of dendritic tree,
sure,
in slice and stuff like that.
So that's highly specific. Though there is also a push, again, some of the same characters who would do uncaging for what's referred to like all optical kinds of setups. So in parallel with using light to stimulate cells, it's very natural as biologists to use microscopes to image tissue, and artificially cells can be made to, when active, change their fluorescence activity. So you basically can record movies that correspond to the activity either indirectly through calcium or directly the voltage, less commonly directly the voltage of the cell. And so you can kind of take movies of the activity of a population of cells, and the sort of dream of all optical experiments that is kind of becoming feasible now is to simultaneously be recording these sort of optical movies of the activity of a whole population of cells that you're recording, and stimulate at single cell resolution a bunch of cells, for example, using optogenetics.
And to be clear, some people actually use the phrase optogenetics to refer to any situation where you're encoding a gene that gives the cell some light-related activity that it didn't have. So if you give the cell the ability to produce light when
or at least change its fluorescence.
Yeah, change its fluorescence, but produce some sort of visual output in that way when it's active, that's also, to some people, a form of optogenetics.
And just to be clear about when we say fluorescence, the reason it's not like producing light, it's like in order for you to be able to image the tissue, you still zap it with a laser and then it emits light. It's just depending on the state of the cell, like depending on the activity level of the cell, the wavelength of light that it emits will be different. So that's kind of how the indicators work at a very high level.
There's also optogenetic tools that were mentioned in some of the reviews we looked at that are not just, say, you can put things other than ion channels into the membrane. So you can have other kind of membrane proteins that are activated by light. So proteins that are coupled to some complex biophysical machinery inside the cell, which is actually kind of like the original thing, whatever those opsins are in the retina.
Yeah, the G protein coupled things.
Yeah. So you can change other biophysical processes in the cells.
Yeah, but those don't seem as common now, but it seems like a lot of people want that to be a direction that this goes, particularly if you could get to kind of changing the expression of different gene levels via light. So you can hit the cell with light and it leads to a complicated process that eventually gets to the nucleus of the cell and is changing gene expression, or uses some mechanism to change gene expression.
That would obviously be there's so many things you could do if you had fine control over this.
Well, that's true. So to me, the reason that optogenetics works, given the level of neuroscience knowledge that people have, the reason it's caught on so much is because it's so simple, right? It is just causing a cell to fire or be inhibited based on the crudest mechanism we know, just directly messing with the ion channels.
Yeah.
And so I think there's kind of a feeling that you can control the cells without having to know too much about them, and it allows us to learn more about them.
Sure.
Many of these other kinds of things are, in a sense, more complicated. You have to know a lot about the cell in order to be able to use those techniques.
Or develop the techniques.
Develop the techniques, but even to do scientific inquiry with kind of long timescale indirect methods, where you're stimulating a cell with an optical tool but it's going to have a very indirect effect.
I mean, I think a lot of people feel like they have some reason to believe that a certain gene is involved in something, and if they want to
turn it
test
on and off during the course of an experiment, that would be really...
Yeah.
So like genes that control, if there was a gene that controlled like synaptic plasticity or something, or I mean, it's conceivable, right? I mean, there are genes that are involved. If you could like temporarily make a certain brain area not plastic or something, or a certain set of neurons not plastic,
you would need to know the time course of the gene expression. So you maybe stimulate the cell with light, wait hours or days or whatever you think to be the case for that.
Yeah,
gene.
but if you're switching to that slow time scale for the course of the experiment, it's not clear that you need light to be the thing that turns it on and off. That was more...
Yeah, no, that would be more for the specificity of the cells that you're hitting probably than anything else.
But you might be able to selectively activate a subtype of cell with a drug that you administer to the animal.
So the question, if you can already genetically identify the cell type you want to mess with, and you also know something about the genetics within that cell that are controlling the process you're interested in, the question is, why do you need to do all this with some opto tool? I don't know.
Yeah, I mean, maybe you need cell type and location or something like that.
Cell type in certain place. Yeah.
It seems
which actually was totally plausible, right? Because it could be like inhibitory neurons, which are everywhere, or like PV expressing inhibitory neurons.
I would say
just want to do it in
some specific brain region. Sure.
There's a lot of situations where it's like for some weird reason you can't do the most straightforward thing, and then people need workarounds. So usually when people use these tools, they're combining it with a bunch of other more standard tools anyways, and it's kind of a hodgepodge just to get the effect that they're aiming for. So what kind of studies have these actually been used in? So one of the interesting things that you can do is that you can put the channels into a cell of interest, and so ordinarily maybe you would have certain cells in the brain expressing these channels, and you would hit them with light and they would become active, and then you could see what happens in other brain regions or to other cells in the same region. But you can also put these channels in the cells and then shine light into an area that you know that those cells project to, and you can stimulate only the cell terminals in that area. So you're studying a very particular projection of cells in one area to another area without the complication of kind of activating all the other areas that brain region might connect to. And so that's something that's been very difficult to do in the past and has been made possible by optogenetics, which can really help dissociate the actual effects of particular connections, because a lot of times kind of everything's connected to everything in complicated ways, and so when you stimulate one brain region, you don't know if you're directly activating another brain region or if there's some kind of loop or something like that intermediate area, and so being able to target specific projections has been very helpful.
There is, of course, this caveat people always mention, right, which is that oftentimes if spikes occur in the axons of neurons, which are the outgoing part of the neuron, then those spikes can quote unquote back propagate to the soma and potentially cause spikes to travel down other axons, which are some other projection that you didn't want to activate. So that's like a problem that can happen.
There's also been experiments that utilize the fine temporal resolution to look at things like things that you consider related to decision making, or kind of like when you're about to do something. There's a lot of areas in the brain that people believe kind of integrate, and the activity kind of goes up slowly over time until it gets to a point where something happens, where the animal, shows its decision by moving a certain way or something like that. And so you can see what happens if you shut down those brain areas at different points throughout that integration. So you can see if it really is, is it just when it gets to this time it makes a decision, or does the neural activity need to be at a certain level? And so if I shut it down early, will it take longer to get to that level? Like shut it off and then turn it, and then let the cells go back on and see kind of how activity recovers, that kind of thing.
And so these are pretty big and central questions in neuroscience, right? I mean, when we don't know how people come to decisions, and there's lots of interest in how brain areas gradually over time integrate information that the organism acquires in order for the brain to decide upon something. And so the temporal specificity here and the brain region specificity here is like that's a very big deal when it comes to trying to tease apart mechanistically what's going on during this integration process. That's like I would say a very kind of major thing that we it was hard. It would have been hard to do without these kinds of tools.
Why can't you do that with electrical stimulation, though?
Well, it would be difficult to target the cells and to turn them off via electrical stimulation. That's something that's kind of specific. So you can turn cells off, as we said, by having different types of ion channels. You can also put the ion channels that would excite a cell into inhibitory cells and just kind of blast the inhibitory cells so that they shut down the excitatory cells that are near them.
So like electrical stimulation, somehow, I don't know why, I guess it's because there's more excitatory cells or something, tends to have been used in a sort of way where you kind of
turn something on.
You kind of typically think of it as turning areas on somehow.
And because it's itself kind of an excitatory, usually the way it's applied, although you could, I guess.
It could be the case that when you stimulate a brain region, you just kind of interfere with its processing.
It's just less known.
Somehow it kind of turns out like
Yeah, so I think there probably have been studies related to this where people did electrical stimulation and just interfered with the integration. But so basically what a lot of people were saying in these review articles is that the first round of experiments that were done with optogenetics was to verify a bunch of stuff that people kind of already thought was true based on older methods. And
more just this, this really enables you to do this kind of, like, in a higher level of resolution, to parse what parts of neuronal circuits are actually implementing what you think they're supposed to be implementing.
Yeah, and I actually just read a paper the other day that used this means of shutting a brain area down by activating the inhibitory cells to kind of test out the role of input to an area versus the recurrent connections in an area in terms of what keeps the activity there going. So you can either turn off the input to that area very quickly and see if that area keeps being active or if its activity goes down, or you can turn off the area itself and kind of use different methods to measure what the impact of the input alone would be to that area. So those kinds of things are interesting. The other thing that I think is really interesting, and this relates to something that people consider kind of a problem still with optogenetics, is this idea that you are still kind of blasting all the cells that you're targeting kind of with uniform activation, or at least if there's differences in activation, it's due to differences in protein expression that you can't control or differences in light levels that you're not intending. So if you're not blasting them all the same way, you're only accidentally not blasting them all the same way. And so really, people believe that the relative patterns of activities in neurons are important. And so you want to be able to kind of control which cells precisely you're turning on and ideally how much you're turning each of them on. And so there's this method where there's something called CFOS, which is an immediate early gene, and basically what it means for neuroscientists is that when you can make it such that when a cell has high levels of activity, it produces this CFOS gene that can then lead the cell to produce channelrhodopsin or whatever ion channel you want it to produce. And so you can make this cascade only be possible for a short window, so you can basically say, I want to label all the cells in this brain region that are active during this particular time with channelrhodopsin. And so now you have a subset of cells that only have channelrhodopsin if they had high levels of activity. And so people have used that to do this really cool experiment where they put an animal in an environment and they saw which cells that represent the environment were active in that environment. So from a whole population that kind of represents environments generally, they saw which ones were active in that particular environment, and they had those cells express channelrhodopsin. And then later on, when the animal was not in that environment, they shined the light on the cells to activate those particular cells, and then also gave an aversive stimulus to the animal, so like a foot shock or something like that. And what this effectively did was kind of make the animal then afraid of that environment, as though they had received the foot shock in that environment.
Can we go through that again? Because this is a really cool experiment, and I didn't quite follow the whole thing. So it's this Tonegawa guy, right? He's at Riken. He's a Nobel Prize winning dude, right? So his lab, yeah. So what's the brain area?
So it's in the dentate gyrus.
Yeah, so they looked at neurons in the dentate gyrus and CA1 or something, which are two different parts of the hippocampus, which is this place that's famously involved in memory, people think, and also in spatial navigation somehow. And those are like the two ways people think of it, the hippocampus. But in this case, they're thinking of it as like a memory-related circuit.
Well, spatial memory specifically.
Yeah. Oh, right, yeah. Spatial memory for different
location.
spatial contexts, right?
Yeah.
As opposed to locations in this case.
Sure.
The difference is whatever that is.
And so in that area, when an animal is in a certain environment, a subset of its cells will be active, and a different subset will be active in a different environment.
Right. So in one environment, they would be in one environment. They would do some manipulation such that the cells during a certain time window, which would be the time window when they're in the environment, will start producing CFOS.
Yeah.
Right? Okay. So so those cells that are kind of specific to that environment are producing CFOS now.
I think they're always producing CFOS when they're active, but they can control the time window where CFOS leads to the production of channelrhodopsin.
So they're always producing CFOS when they're active, but they have this genetic manipulation which says, okay, now the cells that are like active now, i.e. producing CFOS, because this thing is kind of somehow under the control of CFOS in the animal, they start producing channelrhodopsin. So they can kind of label cells that are active at a certain time with channelrhodopsin, make them express channelrhodopsin.
Yeah.
Okay. So it's the like dentate gyrus context A cells, the ones that are in context A, now have channelrhodopsin in them.
Yes.
Okay.
And cells that weren't on in that time don't.
Yeah.
And that's important.
Yeah.
And so then you put the animal not in context A anymore and somewhere else.
Yeah.
And when you shine light on the dentate gyrus, that brain region, the cells that are now producing channelrhodopsin will become active. And the belief is kind of like now the animal thinks it's in that environment in some way, or it's now like open to creating associations that have to do with that environment, because the representation of that environment has been activated in its
so somehow
in its
like
brain.
the memory or something.
Yeah.
Obviously that's kind of a vague notion, right?
And so kind of the normal setup for these kinds of experiments where you test, if you can get an animal to become afraid of an environment, would be that you put it in that environment, you give it a foot shock or something negative, and then you put it back in that environment and see if it freezes or does other kinds of behavior that suggests that it's uncomfortable in that environment. So,
so it's learned to associate the environment with bad things. It's showing so signs of fear now.
Yeah. And so basically they did that experiment, but instead of actually putting the animal in the environment when they gave it the foot shock, they stimulated the cells that represent that environment and gave it a foot shock, and then put it into the environment and could test if it shows signs of fear.
So so
it shows fear in the real environment after association in a different environment, but when stimulated.
Yeah. So it's in a different environment, and they're kind of saying we're going to sort of elicit the memory of environment A in environment B by stimulating, and we're going to pair that kind of evoked memory with a foot shock, say.
Yeah.
Somehow. And then they go back to the actual environment A, and it turns out the previous pairing of the memory of environment A with a negative stimulus is enough to elicit fear when it's actually in environment A.
Yeah.
Which is kind of crazy.
Yeah, and again, there would be really no way to do this without this ability to label the cells that were active and then stimulate those cells only, because that pattern of activity is what represents the environment. You couldn't just stimulate that area because you wouldn't be there wouldn't be an association with a particular environment then.
Yeah, that's really nice.
So a whole nother line of thing that people want to do is potentially use optogenetics for like clinical purposes in a few different settings. So like, if you could, people also, neuroscience research has led to neurosurgeons implanting in people like deep brain stimulators. So like basically electrode systems, kind of like a pacemaker in a certain part of the brain with relatively kind of low resolution stimulation capabilities of an electrical sort. But the success of that for certain kinds of diseases, which we can talk about another time, the success of that has kind of led to some interest in potentially using optogenetic tools as a way of in a more high resolution way, stimulating specific parts of, let's say, a diseased human brain to cause, some benefits to them with respect to some disease. So that's one class of things that would be potentially of interest. Kind of an even simpler thing that people are doing, like more immediately, is using optogenetics in the eye to rehabilitate people who have diseased eyes. So there are various reasons that people go blind. commonly there are common degenerative diseases. So the retina, which is the part of the eye that's photosensitive, is a part of the brain, and that tissue can succumb to many different illnesses. And there are many different ways that people actually want to remedy this. So like there's at least two classes of ways that people want to treat blindness using optogenetics, which, so we're parsing this down a lot, but so one of the ways to treat blindness would be to take the output cells of the retina, which also tile the retina, and cause them to be photosensitive directly.
It's usually the case that there are the rods and cones, which are the photoreceptors of the cell, give input because they are responsive to light. They give input to these other cells that then exit the retina.
Yeah. So
but if you have a disease where the rods and cones are degenerated, then all you have left are these other cells.
These output cells. And there's like a huge amount of processing that goes in before things get to these output cells, these retinal ganglion cells. But basically, the
retina itself is like a fairly complicated neural circuit, which is not obvious to most people.
Yeah, there's a bunch of processing that goes on there. And people study this a lot. So one way, though, is to just like cause these things to be photosensitive and then have the brain kind of learn to process these new totally artificial like light signals from the retina.
Which is not drastically different from like cochlear implants. I mean, those
Sure,
are kind of artificial and people have to learn to process them, and they're still like, not as optimal as the natural ear is, and I think they still have difficulty in that way. So it's in line with other means of rehabilitating people.
Cochlear implants now, they kind of go in, right, and they kind of curl around, and it's a microphone that turns into different frequencies.
Yeah, so they do a decomposition and they like send via electrodes to different parts of the cochlea signals from the sort of sound range that part of the cochlea would most likely have received.
But so there is precedent for people being able to
learn to process what would
kind of slightly
semi-nonsensical signals from the periphery. So that's one thing. My understanding is more people maybe actually think you would use optogenetics to you might actually artificially stimulate retinal ganglion cells with a pattern not from the eye natively receiving them, but you would like put a little optogenetic stimulator in the eye as well or something, and you'd have like a camera. So you'd have like a camera that picks up light outside of your eye, like mounted on your head or something like this.
Yeah.
And you would use optogenetics or just an electrode, people have tried this with just electrodes as well, to artificially stimulate the retinal ganglion cell output layer of the retina. So that
it's not really utilizing the fact that optogenetics is responsive to light because it's using a camera to respond to light, and then the camera turns that into a signal that can go into the retinal
it's just a coincidence that the way you're going to stimulate photosensitive parts of the brain. So like the retina is like a photosensitive part of the brain, and in this case it's just a coincidence that you're going to
unless you do the more hacky thing, which is just put the Channelrhodopsin in, or the whatever opsin
into the retinal ganglion cells and let the brain learn to respond to
figure it out.
It just feels a lot more elegant. It probably doesn't work as well though because you need the pre-processing that the camera could give you or some chip could give you. But
it's an interesting question. It'd be really cool to just do that like in some kind of, people have done this in animals, right? I mean, to some extent. So it'd be really interesting to know behaviorally how much they can just learn. Because that would be fascinating.
I mean, so a lot of it has been done in rodents, which have limited. I mean, it's hard to test their acuity because it's not their main sense. I think so.
I don't know. I just think this is an amusing one.
You could do it in a primate, though, right? Yeah, you can put it in.
One of the papers we read claimed, or something was saying that there isn't a model of retinal degeneration in primates, so they couldn't like fully test its ability to restore.
Because you don't have a primate that has like a messed up.
But it seems like it would be so easy to just kill off rods and cones.
Maybe that's like an ethical thing.
Oh.
So they're not supposed to like gouge out the.
So I do think it's it is amusing to me that there are these kind of there's like one part of the brain basically that's natively responsive to light, the eye. And one way of rehabilitating it would be to use optogenetics to bypass the sort of damaged light-sensitive part of the eye. But sort of, you might also just use optogenetics as like a high-resolution ability to stimulate neurons in a controlled fashion, which kind of gets at a use of computational tools in this case, right? So, if you want to stimulate, in any case, so this could be in the cortex, this could be in the eye. If you have the ability ostensibly to control a whole bunch of neurons, in order to get the neurons to do what you want, you actually need to do more complicated things in some cases than just like
this one
stimulate
on,
them. This one on, this one off. You need to
because they're interacting with each other. So
because they're interacting, you need to kind of have a model.
from what you do to what actually
happens is complicated.
Yeah,
so you need to like kind of have a model of how the neurons might interact or how the neurons will respond to light. And then you need to use kind of standard engineering tools
so this is like
some custom
it's a
engineering.
Control problem, right?
It becomes
Any
a control
engineer
problem.
would be like, obviously you need to have like kind of infinitely fast, infinitely precise control or something to not have to do these kind of things, right?
Yeah. So I mean, in practical settings in engineering, like if you've got a thermostat for a whole building, you don't just like turn the thermostat on when it's too cold. You don't just adjust it and like wait 2 days before the building heats up. You kind of have to predictively use your thermostat. Similarly, when you have a whole bunch of neurons that are interacting, you actually have to kind of predictively know how to play them with guesses as to what the consequences of playing them will be, because you can't do this kind of with infinitely fast feedback.
So this would require
kind of a lot of computational tools.
Modeling and computation of the.
Yeah. So again, in that review, people were citing this idea that kind of we need to understand better or have models that we're trying to test specifically, and then optogenetics needs to advance a bit so that you can more precisely play the piano of neurons, as they say. Yeah. So a question that I have is, and this was asked of other people in the review, is, did this really lead to any breakthroughs in brain understanding, or is it kind of just a refining of the general concepts that we already have? And it seems like most people said, like, at least not yet. There's nothing that's been a big breakthrough.
Have there been any breakthroughs in brain understanding?
Are there breakthroughs in science ever?
Yeah.
I think you just slowly learn new things for the most part.
For the most part.
I mean, some fields are more.
What do you mean? Yeah, what's a breakthrough?
Yeah.
How many breakthroughs have there been?
I mean, I think what's interesting. I mean, maybe we're just naive and in domain, and so our horizons are fairly narrow or something.
Yeah.
But I do think that, and I think a lot of physicists maybe gradually come to this understanding when they study neuroscience for a while. I don't have a physics background, but this seems to be my impression. Neuroscience is just really complex. Biology in general is quite complex, but neuroscience in particular is extremely complex. So it isn't necessarily the case you would expect, like, we're going to have like a 4-parameter model that's going to solve the brain, right? I mean, we know that
the brain has more than 4 parameters. Pretty obvious.
But so in physics, people expect to get kind of elegant, simple, intuitively interpretable, once you know enough math, right? Intuitively interpretable kinds of understandings of things. And I think in neuroscience, there aren't simple conclusions. There's a lot of messiness to it. And so it's not that you would expect a breakthrough. There can be breakthroughs in tools, and I think people consider optogenetics a breakthrough with respect to tools, just like maybe fMRI was before that, or other tools. But, I don't think that you should have sort of, I mean, there clearly can be like big scientific insights, but it's not clear that they would be like
breakthrough.
Yeah. What will it mean for there to be a breakthrough? It would mean to like understand a whole system or something like that.
But I think it's also the case, I mean, usually people talk of breakthroughs and kind of like paradigm shifts as something of a similar family. And what that would require is that we had some kind of strong, we had a paradigm. We had some strongly held belief about how the brain works that could be overturned. And really, it's like, if we found out that glial cells were more important than neurons, that would be that would be a paradigm shift.
Yeah, so that would be a paradigm shift.
That would be a paradigm shift. Or like, there are a few things, but the thing is, like, the things that we know are so basic that we know really concretely that you could call paradigms or something.
Yeah.
If they're wrong, like, we're back to page 0. So that would be just terrible. So like, we should hope that doesn't happen.
Well, but that's always what is hoped when you're in a paradigm.
Yeah, but
so like,
but it doesn't feel like page 0. Like if well, I guess it could be.
Yeah, so I mean,
that's in the scale of the paradigm.
I think the glial example is, I think maybe it's a common example that people come up with when talking about like a neuroscience paradigm. Like, that's kind of one of the only things that people
most of the cells
feel like
in the brain are actually glia, which are these other things.
Yeah,
they're supportive cells.
Thought to be kind of more. They're more like the immune system, or the
structural
vasculature
paradigm. They are
current
supportive
paradigm.
cells.
Yeah. But so like, if it turned out that all of the interesting computations of the brain were really conducted by glia.
Yeah, and like all the neural signaling was like an epiphenomenon.
Yeah.
It was just correlated to the actual computation of it.
That seems to us unlikely. But if that were true, if we somehow definitively found that out.
But I feel like we can't even
But
come up with
optogenetics
many.
isn't gonna do
Yeah, and optogenetics won't do it. But I feel like we can't really come up with any even possible examples of other paradigm shifts because there isn't that many paradigms. There's
So the paradigm is it's neurons. It's like, I don't know.
So like,
spikes being important, like maybe
Yeah.
spikes are an epiphenomenon.
I don't even know.
or
How they're generated, right?
like for example, in computational modeling, people kind of treat
Rates.
like, well, I don't want to get it like though. There's a debates as to how important spike timing versus rates are, but more just that the spiking events are kind of sufficient descriptions of what's going on, and we kind of know that like different neurons release different chemicals, and there can be kind of secondary neurotransmitters that are co-released with sort of when a neuron's active. And so like you can have kind of neuromodulatory effects that are chemically mediated in addition to sort of direct communication from one neuron to another.
The shape of the spike could be important, right?
So so there's like
that
all
would
these,
be very surprising. Yes.
but there's all these very complicated things, and so if those things turn out to be kind of as important as the spiking activity, then that kind of computational simplification would
Break down
a bit.
But wait, so breakthroughs. But yeah, so maybe there aren't any like obvious breakthroughs, and also like scientists don't want to say breakthrough. I mean, it just sounds so ridiculous.
Well,
So-
if you're selling your science,
yeah, okay. Scientists who like are trying to publish Nature papers say that they've made breakthroughs, but they're lying, kind of, or like other people. Whatever. No one's going to say.
It's a bit of exaggeration, or it depends on like breakthroughs can come in many sizes.
Although
you can make a
tools
very
can be
small
breakthroughs.
breakthrough.
A small breakthrough. You break through a small,
Yeah.
weak, feeble barrier.
But it's a
Made
breakthrough nonetheless.
breakthrough. Yeah, I suppose that's right. Yeah, it depends on your perspective what you would consider a breakthrough. If you care very much about something very specific, it might feel like a breakthrough.
If you
I mean, I make
for-
breakthroughs on a
personal
monthly
breakthroughs,
basis
yeah.
on that by that measure.
If you worked on something really tiny for 20 years and then you solve it, that's gonna mean something to you, and you're gonna call
You'll
it.
be solving it at your desk job in some insurance company because you won't have gotten a job. Because science is so horribly competitive. Okay. But yeah, so anyway, the point being, not many scientists are going to be like, oh yeah, there's been 25 breakthroughs because of optogenetics. But like, what are some of the good things? Like that.
Well,
Also, we've covered some of
the memory thing was pretty awesome.
Were you here for this podcast? No.
Oh yeah, sleep. And then there's like the kind of some of the interesting clinical applications.
and then the possibilities for kind of all optical experiments.
Oh, we
Yeah,
didn't
I think
talk
also.
about the idea that you could get rid of pain in humans.
Right.
That's Carl Deiseroth's company's idea, is that because if you're targeting peripheral neurons that are perhaps responsible for chronic pain in certain brain areas, you wouldn't have to go into the brain to turn those off with optogenetics, so you wouldn't have to cut the skull open or worry about anything like that. You could target them at the spinal cord level. So there's a potential for a real clinical application of optogenetics that can come before we solve the messiness of what to do with the skull. So that's optogenetics.
It's a very cool tool, and it's definitely going to be a big part of neuroscience for quite a while, it seems like.
Yeah.
Yeah.
Wow, that was such an anticlimax. God.
Till next time.